Thank you so much. Please be seated. For a mind that knows is a mind that is truly free. Distinguished guests, ladies and gentlemen, professors, distinguished elders, teachers in the audience, deans and directors, chief medical director and representative, all members of the academia that are here, they are distinguished fellows of the Nigerian Academy of Science, quite a number in this hall, members of the family of the Olumbe Basir, the late Professor Olumbe Basir, a distinguished icon. Ladies and gentlemen, without forgetting the men of the press and the information technology unit anchoring this, members of the foundation and staff. I welcome you once again, and permit me to invite to give the welcome address the chairman of the board of trustees of the Thomas Basir Biomedical Foundation. Is ably represented by the 11th provost of the College of Medicine, a distinguished fellow of the Academy of Science, Professor Oluwabumi Olapade Olaopa.
The chairman of this occasion, the Vice Chancellor of the University of Ibadan, ably represented by the Deputy Vice Chancellor Administration, Professor Ayola, the family of the late Professor Olumbe Basir, after whom this lecture was named, the guest lecturer, our distinguished Professor Christian Happy, who is one of us. He should really need no introduction to the University of Ibadan. Christian, as we all know, is distinguished himself and has won many laurels. And it's therefore, for us, a very great occasion that he has agreed to give this lecture, which we are sure we would all find very informative. As we have been told, this lecture is being streamed online, and we acknowledge those who have joined us online. The chairman of the Thomas Basir Biomedical Foundation is Professor Ladele Kale, who is unable to join us today. He hopes he might be able to join us at a later event, but he couldn't make this morning. And he sends his regards to us all, distinguished ladies and gentlemen. The Thomas Basir Biomedical Foundation was started in 1974 by the late Professor Horatio Olusegun Thomas, who is a former Vice Chancellor of the University of Ibadan, at a time when it was called the Wellcome Foundation. And the whole idea at the time was to support biomedical research. Professor Thomas recognized that young researchers in particular were struggling with getting grants to undertake their, what was seemingly small research projects. So he was quite keen for this group of academicians to be built up both in grant writing, but also to get their research projects off the ground. So he donated his own share, family share of the Wellcome Trust, to the Wellcome Foundation to support young researchers, mostly postgraduate students, but also young faculty. And the idea was capacity building in-house, so that we do not always have to have a young researcher competing with the likes of Professor Christian Happy and Professor Eruka Okeke for NIH grants. And the other important aspect of the foundation was that he thought it would be a very good forum whereby we could discuss research findings in our own environment that had been sponsored by our own organizations and therefore build a community of practice which will lead to the ownership of our own research ideas and therefore research results. I'm pleased to say that nearly 50 years after the foundation was started on this initiative by Professor Thomas, it has transmuted to the Thomas Basia Foundation after the fact that Professor Olumbe Basia, whose lecture we're giving today, was actually the chairman of the Board of Trustees from 1979 to 2001. And we have strived to maintain this ethos. The foundation has given a lot of grants to young researchers, and our other aspect are lectures like this where we bring the big giants. We alternate between the Basia lecture and the Research Genomic lecture. And we bring giants to give us an insight into what they are doing and to stimulate our young researchers to see what they should aim for. Today, I'm sure we're going to be treated to a diet that we would all be very happy with. No pun intended to the name of our guest lecturer. And although vote of thanks will be given later, I must thank the Vice Chancellor, Professor K.O. Adebowale, for honoring our invitation and for asking the Deputy Vice Chancellor Administration to represent him, even though he's away. And of course, I must thank the Deputy Vice Chancellor Admin for attending. He was the first, second to be here. Professor Atinmo was the first person to arrive. And we thank everyone who has come. As we've said, we recognize all our senior professors, deans and directors, former deans and directors, and I hope we enjoy our lecture. I must not sit down without saying that could you please encourage for the senior members of academia, your research students to apply for the Thomas Basia Foundation grants. We give anything from 250 to 1,000 to 1 million to support research that is being supervised by an eminent researcher. And all we have to do is a 3-page application that will show that this is a research that is worth supporting. And by and large, we do give sufficient numbers of grants every year and intend to continue to do so. We also acknowledge SKG Pharma, who continue to support us. Thank you very much.
Thank you so much, Professor Oluwabumi Olakwade Olaokwa. Distinguished guests, ladies and gentlemen, it is my pleasure to invite to deliver the chairman's opening remark, the Vice Chancellor of the Premier University, who is ably represented by the Deputy Vice Chancellor Administration, Professor Ezekiel Ayola.
Thank you very much. Our 11th Provost College of Medicine, Professor Bumi Olaokwa, the distinguished guest lecturer of today, Professor Christian Happy, Professor Sola Botonshaw, and other members of the high tables, distinguished ladies and professors that are here stated, our teachers that are here. I can recognize Professor Ekundayo, who is there. You are recognized, you are welcome. So, ladies and gentlemen, both here seated and online, it is my pleasure to be here this morning to represent our 13th Vice Chancellor, Professor Kayode Adebowale, FAS MI9, who is not around, not present in Ibadan at this moment. He has traveled outside Ibadan on official duty, and he has asked me to deliver this message. It's a written message from him that I am going to deliver now. I warmly welcome you all to another in the series of distinguished fellow lecture organized by the Thomas Basia Biomedical Foundation. Permit me to congratulate the chairman Thomas Bastia Biomedical Foundation, Professor Oladele Kale, FAX, and members of the Board of Trustees on the occasions of the 2022 Professor Olumbe Bastia Distinguished Fellowship Lecture. It is a great feat, and the University of Ibadan is indeed proud of your contribution to promoting biomedical research and building research capacity in human and veterinary medicine and pharmacy. The 2022 Professor Olumbe Bastia Distinguished Fellow, Professor Christian Happy, is also one of our own, having been trained in our great University of Ibadan. He has made outstanding contribution to the field of genomics, of infectious diseases and human genomics, and won many laurels. Professor Christian Happy, I congratulate you on your nomination by the Thomas Bastia Medical Foundation as the 2022 Olumbe Bastia Distinguished Fellow. The late Professor Olumbe Bastia was a professor of biochemistry in this great institution, the former head of the Department of Biochemistry and Dean of the Faculty of Science. He was a researcher par excellence who made significant contribution in the field of biochemistry. He also served as chairman of the Thomas Bastia Biomedical Foundation from 1979 till the year 2001, when he passed on. Okay. When he passed on to glory. It is encouraging that his contribution to science and research are recognized and his name is being immortalized. The Thomas Bastia Biomedical Foundation seeks to promote human and animal health for the benefit of the Nigerian people by advancing medical knowledge through biomedical research. The foundation has been able to achieve its aim through one, the direct award of grants to graduate students, doctoral fellow, scientists, such as human and medicine within and outside the University of Ibadan. Number two, disseminations of new findings in medical research by organizing roundtable discussion, seminars, conferences, local publishing reports, proceeding information. Number three, the administration of the nation and developing thought and the members here fellowship and lecture or award and awarding organization. We are addressing today. Foundation is distinguished lectures forum for one for the distinguished fellow. This is our personal achievement in research to illustrate the importance of health research. But for advocacy of imperative that the Nigerian government and citizens must recognize the need to support health research. Several doctors and lecturers have been recipients of the Thomas Bastia Biomedical Foundation grants. I appreciate the Thomas Bastia Biomedical Foundation for the strong support of several proposals which has helped to build research capacity in the field of human and veterinary medicine and pharmacy over the years. The foundation is constructing a Thomas Bastia Biomedical Foundation building which will house a 40-bedroom postgraduate hall at Ajibode. This will certainly be a valuable resource for postgraduate students of the University of Ibadan and will ease the accommodation challenges encountered by these students. Once again, I welcome you all and heartily congratulate the distinguished fellow of today and the Thomas Bastia Biomedical Foundation. Thank you. Thank you very much.
Mr. Vice Chancellor, sir, thank you so much. Another round of applause for the Deputy Vice Chancellor Administration, is standing in for the Vice Chancellor. I feel highly honored to be here today because I can see many of my teachers and mentors, distinguished professors of the University of Ibadan, and to give the citation of the guest lecturer is a fellow of the Nigerian Academy of Science, a lady who was recently announced as a 2023 winner of the Microbiology Society Prize, a prize that is awarded to global leaders in the field of microbiology with impact far beyond their reach. One of the nation's most cited researcher, and one of those with the highest number of grants attracted to the University of Ibadan, Professor Iruka Okeke.
Thank you for the kind introduction, Dr. Lawal. And thank you all of you who are attending here in the room and online. It's indeed a pleasure and an immense honor to be able to read a very abridged citation of Professor Christian T. Happy. May I ask Professor Happy to please rise? Thank you. So Professor Christian T. Happy is a professor of molecular biology and genomics and director of the World Bank-funded African Center for Excellence in Genomics of Infectious Diseases, ACEGED, in Redeemer University, Ede, Osun State, Nigeria. Following his first degree in biochemistry at the University of Yaoundé in Cameroon, he obtained master's and doctoral degrees from here in the University of Ibadan in molecular parasitology. He completed his postdoc training at Harvard University in molecular biology and genomics of malaria in 2003, and he subsequently worked at Harvard University as a research scientist and became an adjunct professor there. From 2004 to 2011, he was also a scientist at the Malaria Research Group, IMRAT, College of Medicine, University of Ibadan. Professor Happy's research focus is on infectious pathogen and human genomics. He has worked on a range of pathogens across the tenure of his career. His work on malaria genomics revealed molecular markers of drug resistance to anti-malarial drugs in Nigeria and contributed significantly to the national policy change that happened in 2005 that changed the first-line treatments for anti-malarial drugs in this country. In 2007, Professor Happy's work made it possible to diagnose Lassa fever in Nigeria at the Irua Specialist Hospital in Edo State, Nigeria, on site. And it has since led to the development of a 5-minute point of care test for Lassa fever. His cutting-edge Lassa fever genomics research has also enabled tracing ancient origins of this virus to present-day Nigeria and mapped its spread across West Africa over the last 400 years. Early endemic area research in Lassa fever genomics has accelerated the development of Lassa fever vaccines, among which 2 are currently undergoing clinical trials. He and his team have used genome-wide association studies to identify loci that make some individuals resistant to Lassa fever infections, making it possible for us to start figuring out the best ways to protect people from this disease. Professor Happy and his team members diagnosed the first case of Ebola virus disease during the dreaded 2014 West Africa Ebola outbreak, the largest Ebola outbreak that has occurred to date. Professor Happy's colleagues used genomics and deep sequencing to sequence the first 99 Ebola genomes during that outbreak and made the data openly accessible to the international scientific and research community. This is now the textbook example of the use of genomic epidemiology to facilitate outbreak response in real time. And it is a model that has been applied currently in the COVID-19 pandemic. Professor Happy and his team used translational genomics to develop the first rapid diagnostic tests against Ebola virus disease within 6 months of the West Africa Ebola outbreak in 2014 to 2016. And the test was the first rapid test approved by the WHO and the US FDA for emergency use authorization. Professor Happy's work has made it possible for our National Public Health Institute to respond rapidly and in a focused way to infectious disease threats. In 2018, he used next-generation sequencing to discover a new yellow fever strain that was responsible for what was then considered a mysterious viral hemorrhagic fever outbreak in Edo State, Nigeria. This information was passed to NCDC, who declared an outbreak and successfully contained it, saving thousands of lives. His group has also discovered 2 novel viruses, EKV1 and EKV2, in Ekpoma, Edo State, using, again, cutting-edge technology, in this case, microbial metagenomics. We talk about the need for pandemic preparedness. Instead, we're still talking about it in the midst of a pandemic. But ACEGID was unusual in that it was actually prepared for COVID-19. Within 48 hours of receiving samples of the first index case in Nigeria, Professor Happy and his team had sequenced the genome. They have since sequenced over 5,000 SARS-CoV-2 genomes in Nigeria alone and helped to coordinate an unprecedented sequencing activity across our continent. And discovered the B.1.525 variant of interest in Nigeria. In fact, ACEGID has been instrumental in being able to map the entrance of every single variant of interest and concern in this pandemic. The seminal work on COVID-19 genomics has not only provided insight into genetic maps, origin, evolution, transmission, and spread of coronavirus in Nigeria and Africa, but also paved the way to developing new CRISPR-based diagnostic tests for COVID. This test has been approved by the US FDA and is used in the USA for COVID-19 testing. Their genomic sequencing work has also resulted in the development of a COVID-19 vaccine candidate called COVID-Diovax in collaboration with the University of Cambridge in the United Kingdom, which is now in clinical trials. Christian Happy is a major contributor to the 1,000 Human Genome Project and referenced genetic variations of the human genome. And he's actually one of the leaders of activities on the African continent to yield to the sequencing of our own local human genomes. Professor Happy is a member of several international advisory boards. Since we're all itching to hear his lecture, I'm not going to go through the long list here today, but other than to say that it is very impressive and only surpassed by the number of funding agencies and private foundations that are funding and supporting the work by his institute. If you haven't read his papers, get on PubMed now, search his name, read his papers. They are in journals like Science, Nature, the New England Journal of Medicine, the top journals in the biomedical field. He's won several prestigious national and international awards. Some of the more recent ones include the 2020 Bailey K. Ashford Medal of the American Society for Tropical Medicine and Hygiene for lifetime and impactful contribution to infectious disease and tropical medicine. He's also the 2021 winner, he and ACEGID are the 2021 winners of the Gold Medical Prize Award of the Nigerian Academy of Science and the 2022 Nigerian Healthcare Excellence Award for the most outstanding genomic laboratory of the year. Professor Happy is a founding fellow of the Academy of Medicine Specialties of Nigeria, FAMS. He's also a fellow of the Nigerian Academy of Science. He's an exceptional mentor and advisor to almost all groups on the continent that are performing pathogen genomics. He embodies passion, intellectual drive, innovative spirit, all the things that African science needs to address the many challenges on our continent. His scientific contributions have gained him the admiration and respect of researchers worldwide in virology and in global health. Professor Happy.
Thank you, Dr. Iruka, for that presentation. It is a pleasure for me to be here today and to give this lecture. Before coming here today, I asked my colleague and friend with whom I spent many, many years working, Professor Botosho. So what was going to be the audience? And she told me academic members of the community and then graduate students. And I said, that is a great thing. I want to use this platform to thank again Professor Botosho for not being a wonderful colleague, but also to being one of my teachers. And she taught me some great stuff. She used to be my Yoruba teacher. She taught me all the good Yoruba that I know, the good ones. Everything good about Yoruba that I know, she come from her. But the bad ones come from Mechanic Village where I used to go. And from Danfo drivers and Motorway conductors. That she's been a fantastic companion all over the years. And, you know, again, I want to use the opportunity to say thank you for good companionship because you've been wonderful. So today, I won't talk about malaria, but let me talk about some of these things that is post-malaria. But I also want to put it on record that when I started this work, you were part of it. We started, and then there was a big struggle. And then I want to send this message to the young ones. As we heard, I am not a virologist. I'm a biochemist. I was a trained biochemist who put this step in parasitology and eventually veered into the field of virology. So, the message to the young ones is, think without border. When we started this virology work, in this same university, some people thought that the field of virology was their property, personal property, and as such, nobody should dare go near it. Nobody should think about it. But my message here today for the young ones is, think without border. Let nobody tell you that you cannot do anything. Let that professor that who tells you that, look, my father is the owner of one particular field. Agree with him, but think differently. And go ahead and then do what you think is good for you and be inquisitive. So my message today is, young ones, think without border. Do not be restrained by your area of specialization or your field. Do not be restrained by the fact that they call you either a pharmacologist or a microbiologist or whatever. You can do beyond what you are being trained to do. So think without border. So, I will talk about genomics today and then mostly related to viruses. So, and I just want to mention here that, full disclosure, I direct this center, and basically the center is one of the World Health Organization and Africa CDC reference, one of the two reference centers for genomic infectious disease, genomic research and innovation on the continent. There are two centers, reference centers, one here, our center, and the other one in South Africa. We're also a center for the World Bank, which is one of their centers of excellence. Now, this is my outline. I will scan through this very quickly. So I will talk about genomic epidemiology, but most important, I will talk about translational pathogen genomics. So which is not just to do genomics, but how to show how we are taking genomic beyond just looking at genes and genomes, but translating that into product, point of care diagnostics, and then products that affect positively or impact positively, public health response to epidemics. And I will take a few examples to illustrate this. I will also talk about how we use genomics for pathogen discovery. And I will be closing by mentioning this program called Sentinel that we set up before the COVID-19 pandemic. And this is an early warning system for pandemic preemption and response. And we demonstrate how this program has been able to positively impact not only Nigeria, but also the African continent, and absolutely show how this program directed the response of COVID-19 in Africa, and is in many ways responsible to how Africa averted this pandemic, in a way that was not done. And finally, I will close in by showing the whole component of capacity building, because I know this foundation has a lot to do with capacity building. So this program is all about capacity building and training, and I believe that that's where the future of Africa lies. So, you hear about genomics, and then we understand what is genomics. It's that science that harnesses knowledge of the entirety of genes of an organism and use it actually to do a lot of things. And among those things could be, diagnostic treatment, developing novel countermeasures. But the genomic revolution has changed the way, healthcare and research is being conducted today. For instance, today you're hearing a lot about personalized medicine, and that's where we're going. Today, we are not treating people, the treatment is no longer on the basis of generic treatment, but it's actually based on your genetic makeup. Some diagnoses are actually made based on your genetic makeup, and some vaccines and even therapies are actually made on your genomic makeup. But a few years back, we realized that this technology, or this new science called genomics, has accrued so much in terms of economic benefit, health benefit, and knowledge to the global north. And yet, Africa was not participating into this new revolution. And that resulted in a huge divide between the rest of the world and Africa. And then the only reason why Africa wasn't contributing to this revolution was simply because we were not prepared. We didn't have the knowledge, we didn't have the skills, we didn't have the infrastructure, and as such, it became, a major thing, a major problem for the continent. Some of us felt that, this genomic revolution could be the equalizer. We've been behind for many centuries, but we believe that with genomics, we can actually be, catch up and actually level the field, because the richness of the African genomes and the richness of the African biodiversity. So this could become a very big equalizer. And we found that, if we were able to, quickly move up and share knowledge and educate our people, we should be able to actually move fast and even ahead of those that think that they're actually the custodian of this knowledge, by showing them that we can actually harness this knowledge and then do better than them. So, and if you look at this map, we decided to focus on something that is very important. This is a global map. But what actually makes Africa very special when you look at the global map? Of course, African slaves have been taken all over the world to build the rest of the world, and Africa remained very poor. But there is something that Africa has that the rest of the world doesn't have. And that is exactly what you see here, right? You could see here across the globe, as they're coming down, these are all pathogens with pandemic and epidemic potential. But if you look at the concentration, they are mostly concentrated in Africa. And studies actually show that 70% of the pathogens with pandemic potential or epidemic potential are concentrated in Africa. So it makes us believe and understand that God has been very wonderful. He's been very wonderful creating an Africa that is rich in natural resources, rich in minerals, rich in human resources, but also gave Africa something that was unique, a very powerful biodiversity. And as part of that biodiversity are viruses, are pathogens, and parasites. Therefore, it should be considered as a source of wealth for the continent. So therefore, Africa should harness these pathogens and turn this into wealth. For those that have worked with me in the past, anytime you see something like this or we see problems, I don't consider this as problem. We never saw this as problem. We believe that all these pathogens that you see concentrated in Africa are actually, opportunities in disguise, because they could be used actually to teach the world how to do good science, to teach the world how to make discoveries, and actually to give Africa its rightful place in the communities of nations. Because we have this as part of our biodiversity. We should be the custodian of the knowledge. We should be the one driving the science when it comes to pathogen genomics or pathogen discoveries or developing countermeasures. And as such, we felt that through the genomic technology, we could actually harness one of our strengths, which is thought to be a weakness, which is the viruses and then the parasites circulating on the continent, to make the continent, a powerhouse for, new knowledge and also developing novel technologies toward a global public health. So, and then we created this platform, which is made of academic institutions within and clinical testing centers in Africa, and then with international partners. And then it has two goals. One was educational. And that educational goal is really built around creating, this environment that transcends national boundaries, and where people can come and then do what I call high-quality translational genomic research for health development in Africa. And by creating such an environment, specifically with belief that, through that, we could basically train a critical mass of well-trained African scientists that can harness knowledge and the skills and the tools of genomics toward elimination and control of infectious diseases. But to do that, we needed to create curricula. In order to infuse knowledge into people, you needed to create curricula. And we set up the very first molecular biology and genomic program geared toward infectious diseases on the African continent. And this program received an international accreditation through the AQES, which is a European, accrediting body, and that really, has put us apart with any other institutions in Europe, whereby our graduate students with masters and PhD in this field are equivalent to any European Union, a graduate student. So, but then we basically just not only did this, we basically also just ensure that we're engaging communities in public health prevention, and effort. So which means we're not just working in the ivory tower, we're not working in the university, we're taking this knowledge out, we're translating this knowledge in solving public health problems. You're going to be seeing that in the next few minutes. But then around the training, you really want to have a training program that is driven by research. So the question is, what is our research training program that really is very important and that could be very catalytic for the African continent? And one of the things that we saw is the fact that, we needed to focus on something that is very peculiar to this continent and something that can cut across the continent. And we identify febrile illness as one of those problems. And identification of febrile illness became very central and core to our problem, because we know that most infections or most diseases, when they start, they always start with fever. In our own environment, in those days, we used to say IBA, that is malaria fever. But then when you have malaria fever and it's not malaria, then it is something else. In our own context here, we often say that after malaria is what is typhoid. And if it's not typhoid, then we start creating some other suspect. But because our doctors are very ingenious, often when you come here, you go to the hospital, they will tell you that you have malaria typhoid. So they bombard you with anti-malaria drug, and then they give you antibiotics. All of this is simply because we do not have the ability to actually detect quickly what pathogen is responsible for fever. So our program really was to focus and identify all of the parasites or all of the pathogens that are driving febrile illness across, not only in West Africa, but across the continent. In doing that, we basically establish what we call the microbial landscape. If you understand and establish a microbial landscape, then you can develop the next generation diagnostics, you can develop the next generation therapeutics, and then you can also develop the next generation vaccine. So, and that's basically what this program is all about. But in order to do that, we didn't want to do business as usual. We really wanted to start using novel technologies and innovation to drive this program. And that's the reason why we started using genomics, and in this case, using microbial metagenomics as a way to have a deep dive into the samples or into understanding what are the key drivers of febrile illnesses. ultimately, by bringing the research and education together, we basically are meeting one of the Africa critical needs, which is genomics research and in genomic research and education, and that has to do with capacity building. You really need to build a critical, capacity in order to actually to be part of the genomic revolution. I will believe that through that, we basically, through this program, we've been able to set up some genomic sequencing facilities, diagnostic core labs, and then also sustainable science career for young students. So the idea is not just to train, but is also what is the, the academic, or what is what I call the path for, development for anyone that you're training. So you see that through our program, we've also set up a platform where students can evolve from being a student to be workers and then to actually earn what I call decent salaries. But ultimately for us, the goal is to build the next generation of African pathogen hunters. That is basically training the young people and making them hunters so that they can actually, hunt and catch these viruses, catch these pathogens that actually harm us. You agree with me that one of the weakest, link of the global disease surveillance program is actually the detection. So our inability to detect pathogens early is actually what is making us play what I call defense. We are always very reactive. But with the skills and the knowledge that we have today, I think we can change the tide, and then we can start playing offense. Basically, getting them, characterizing them, and then developing the countermeasures, early enough before they come to us. And we really set up this program and demonstrated that it could work. And our first demonstration really came in 2014, where we really changed the way the global health community respond to epidemic. When we had Ebola outbreak in West Africa in 2014, as far back as November 2013, when it started in Guinea and the world was looking away, I was very scared. I was very afraid. Because Ebola often happened in places in the Central African Republic that are dense, that are sparse, with sparse population. But in West Africa, we seem to have populations that are very dense. And the question is, if you have a disease like Ebola, happening and spreading, then we're all in trouble. So what happened at the time is the fact that when this happened in Guinea, we really thought about, start thinking about using genomics as a way to respond. And as far back as April 2014, I got a group of friends and colleagues, and then I expressed my worry and my concern about this. And we identified diagnostics as one of the major weak points if we were ever going to be responding to this epidemic. And of course, if you cannot diagnose a disease, you cannot respond. And that for us became a focal problem. We went on to train a few. Hello? So we went on to prepare the ground and train people in two countries where we were doing work before, Sierra Leone and Nigeria. And as we rightly predicted, around May 20th, 2014, we had the very first suspected case in Sierra Leone. And one of the young men that we trained, Augustine Gelba, was the one to detect that, to confirm that. But interestingly, within the first week, we had about 35 suspected cases, and we confirmed 14. But what was interesting is at the bottom of it, or at the center of it all, was a traditional healer who thought he had the power to cure Ebola, and had patients coming to him, and he became that person through which all cases in Sierra Leone actually were being contracted. He died along the line, but before dying, he had actually created a lot of havoc. So, and this for us became very important. You can imagine within a week, 35 suspected case confirmed 14, and this information were actually shared immediately with public health authorities in that country. But what we did that was unprecedented is the fact that in the first 2 weeks that followed that first week, we generated the first 99 full genome of Ebola virus in Sierra Leone. But we didn't keep the data to ourselves. We didn't think about using it for publication. We made the data openly accessible and openly available to the international research community. The reason for us was we were running for solution. we're speeding toward getting the right solution and get to demonstrate the use of genomics, not only to actually to sequence these genomes, but actually to use the information to help public health response and help the country to respond to the outbreak. We generated this method that we actually shared, but most critically is the fact that we changed the way sequencing was done. It used to take about a month to actually go from sample collection to depositing this in gene bank. But because of the situation, we were very innovative. We created novel ways of approaching things and doing it. And we were able to, here, from sample collection to submitting the data in gene bank, 10 days. So when people ask, how did you do sequence COVID in Nigeria or the other things in 48 hours? I said, I built on the fact that we started by cutting down, the time by 3/4 during the Ebola outbreak, but keep improving on the technologies to the point where you can do it in 24 hours or 48 hours. But now I can tell you we can do that in 24 hours because we've actually cut back again. And that really shows the relevance of real-time genomics when it comes to responding to epidemics. So we made this openly accessible and openly available. But then this really gives you that great understanding. What the world is using today for COVID-19 response is a carbon copy of what we did in 2014. We did genomic sequencing in real time. We released the data in real time. We created an open data access. So what you see GSED is doing today is things that we started in 2014. And that for us, again, is preaching to what we said before, which is Africa has what it takes to lead. Africa has lessons to teach the rest of the world when it comes to things that are part of our biodiversity, and these viruses are. So we made all of these things available, and our paper in Science was not published until 8 months later. So you can imagine at that time being that bold and daring. You generate data in real time, you make it available. You're not thinking about publishing. You just think about publishing 8 months later. And that becomes the example that the world is using today. But this here, for instance, is a very clear demonstration at the time how we were able to, first of all, show in the case of Sierra Leone, where you have an epidemic, you don't know what is going on in the country. But in the case of Sierra Leone, we showed here that there are 4 different variants that entered the country at the time. And you could see at different periods, you could see SL1, 2, 3, these are different variants. But by June 16, we observed that one particular variant, which is SL3, had taken over. How would a variant, you have several variants that are circulating during an epidemic, and one of them takes over and becomes responsible for spreading the transmission of like wildfire. So the question was, what is responsible? So we went on to actually look into these viruses, and then we saw accumulation of mutations across the virus, over 300 mutations, but by the time we looked at the viruses critically, and those that were really responsible for severe disease, we realized that there were 21 mutations, very, very, that were consistent. They have become fixated, and those mutations were responsible for what was responsible for the increased transmissibility. So this was the first time during an outbreak where you could use genomics to show that one particular variant has taken over, and what is responsible for taking over or increase its transmissibility is accumulation between a particular, particular aid of the virus that is responsible for facilitating the virus to be more efficient in entering the human cell. And while we were doing this, the anecdote around this is we were dealing with an international organization called the World Health Organization, which at the time, of course, was founded in 1945, has been operating with systems built in 1945. They couldn't understand that we were doing this during an outbreak and using it to respond to an epidemic. For them, we were doing research, and research is not allowed during an outbreak, simply because the modus operandi of WHO at the time was when there is an outbreak somewhere, they bring the so-called expert in court. They will come and they work with the local people with little or no understanding of the cultural and then the national context, go back and take all the samples to some lab in Europe, and then they will be telling you 2 years later what was responsible for that epidemic in Africa at the time. But we told them at the time, I remember very vividly, telling them that they are 50 years behind, knowledge. And therefore, they should understand that we are operating at a level that they don't understand. And this, we're able to convince the president of Sierra Leone, because we will see that at the end of the day, we understood the value of this technology because they used to give him daily report. And using this to identify hotspots, then the country was able to go through. But apart from this, you will see here in this map how we've been able to use genome sequence data to show transmission between individuals, between communities, and between towns and cities and villages, and then even across country. So this, for instance, if you look at it, this is space, and then this is time, and then this is space. And these are using just genomic sequences to show transmission patterns. This is between communities, countries, and eventually crossing the region. And this really shows the power and the firepower behind this approach and this technology. So you don't have to be a rocket scientist or be a genomist to actually use information like this to actually see exactly where the hotspot is and develop, the policy to contain that area to perform like things are locked down and avoid people moving around with disease. Oh, that's true. Well, let me try. I think it works. Huh? So we basically just translated this into point of care, and then this became the first generation Ebola RDT that was approved by the European Union, the WHO, and then the US FDA under emergency use authorization. The introduction of these tests technically bent the curve and flattened, not only flattened, but bent the curve of this outbreak, because the doctors and the lab people could actually get the result within 10 minutes. We had a situation whereby people with malaria fever were going to the Ebola treatment center or diagnostic center, and then they got exposed to Ebola and then took Ebola back. And with this test, we didn't have to wait for 4 days or 6 days to get your PCR test. You could get your result within hours. What is good is that this test was as good as PCR because we did a neck-to-neck analysis with PCR, and we had a sensitivity pretty to 98%, and our specificity was 100% compared to PCR. It's not very clear here, but I just brought this picture up because this is a gathering at Redeemed Christian Church of God. My lab was at Redemption Camp at the time, and then this is the kind of gathering that we used to have on a Friday, and this was in the middle of the outbreak in Sierra Leone, and then we were very scared of Ebola spreading over to Nigeria. And when you look at a crowd like this one, it's about 3 million people gathered in one single place. And what comes to your mind is that what will happen to a country like Nigeria if you have a few people in the 3 million crowd having Ebola? It was going to spread like a tidal wave. It was going to spread like wildfire. So that for us became very, very, scary. And we got prepared for it. And I do remember on July 21, or July 20th, 2014, I got that chilling phone call from Professor Oyewale Tomori, calling me at 11:40 p.m. And he told me, Christian, I understand that there is a suspected case of Ebola in Lagos. That the person landed, the whole place is in disarray. We seem not to know whether it is Ebola or not because this person, all of the tests have proved negative. He's coming from Liberia. And what he told me at that night is, you have to go and then look into this. And I left my house around 9 p.m. that night and drove to Redemption Camp. The sample, as I was driving, the sample was leaving Lagos to the lab, and we met there and worked. I worked all night with a colleague of mine in conditions that are not ideal. We're talking about using an Ebola sample in a laminar flow hood, the type of hood that I used to grow malaria culture. Very risky, but we're committed to do it. And in the morning, we were able to actually confirm the fact that we had the very first case of Ebola in Nigeria. And guess what? Within the next few months, we were able to work with Nigeria public health authorities and we were able to contain Ebola. What makes Nigeria so different from others is the fact that there was a very strong collaboration. The sample used to get to us in real time, we analyze and then send the result back, to the emergency operation center, and then they know what to do. Nigeria was able to conquer Ebola in 93 days with only 20 cases and 8 deaths. That is unprecedented in the history of public health. But it is cooperation. You could see here, for instance, these are genome sequence data, and then these are contact tracing data. You realize that these actually were superimposable. And that was the first, again, in this country, where you could use genomics as a way to guide public health, showing exactly to public health authorities where to go and what to do. Now let me change gear a little bit and then talk about a disease called Lassa fever. A disease described in Nigeria in 1969 and that killed a few American nurses. And in 2007, I heard about that disease for the first time. I have to be very honest that before 2007, I'd never heard about the word Lassa fever virus. I was at Harvard at the time doing work and reading, online newspapers about Nigeria. You hear about Nigeria, Lassa fever killing 50 in Edo State, Lassa fever killing some people. And I became interested, and then I was talking to a friend of mine too who probably was interested in that. And I remember coming back, was it January 2008? And then I met Professor Botosha, and I was telling her, I have to go to Irua. I told her I have to go to Irua, and she was saying, Irua, what is going on there? Why, what is so interesting about Irua? I said, no, I heard about the disease, Lassa fever. He said, what are you doing with Lassa fever? I said, look, let me just go and see what is going on. And I went to this village called Irua Specialty Teaching Hospital. By the way, this tells you a bit about the virus, but I don't want to bother about it. I went to this village, and then what I saw, it was a bush. It was a bush, and in that place, they used to lose medical personnel. When the government would transfer medical personnel there, they would not go. It was like a burial ground. People would not come to the hospital because they knew that when you go there, you will eventually die. There was no diagnostic, there was no way of treating anybody. And yet this disease used to kill people in number, in thousands. So the first thing we did was to work with the folks there and establish a small, I call this a small cubicle. So it's a 4-wall thing with partition inside, and that's what we set up. And I came back 6 months later with all the full complement of equipment, and we set up a lab there. And on this famous day, we basically just did the first molecular diagnosis of Lassa fever in Nigeria. The difference this diagnosis started making is the fact that anytime they had an outbreak, sample used to be sent to Europe. And they would get the result about a month later. And within that period, 90% of the patient would have died. And yet with supporting treatment, they could have actually saved a few of them. We went from there to actually work with them to set up a clinical treatment center. But then this is a lab that we recently built there. So we went from this little cubicle to have what I call state-of-the-art facility with all full complement of genomic facility. So in this lab, they do everything from diagnostic to sequencing from this little bush. So, of course, it is good to set up diagnostics, but then diagnostics shouldn't resolve the problem. How do we accelerate the path toward vaccine development? So what we did was basically to, first of all, engage in sequencing the genome. So if you sequence the genome, then you understand you can identify the epitopes, and through those epitopes, you could quickly develop a vaccine. And that's what we did. By going on, we beat the very first genome sequence of the Ebola, of the Lassa fever virus, using unbiased, sequencing, and we generated the very first genome of this virus. Generating the very first genome of the virus actually did two things. We went from using PCR tests to creating a molecular diagnostic test. And we did it not only in Nigeria, but we're also doing it across in Sierra Leone. But along the line, we were basically trying to answer some very fundamental questions. And one of those were, what is the origin of this virus? If it was described in Nigeria in the 1969. And our findings were very clear. This virus emerged from present-day Nigeria about 1,185 years ago, to be more precise. Was circulating within Nigeria for 600 years, and then moved out of Nigeria about 400 years toward West Africa, and then got here. You can imagine, along the line you see there are changes. So you have lineages 1, 2, and 3 in Nigeria, but by the time it comes over here, it has changed, and then you have only lineage 4. We're able to develop a rapid diagnostic test very quickly for Sierra Leone because they had only one lineage. That, within a year, we had the diagnostics. But I remember for Nigeria, we had a lot of problems. We didn't know at the time that we were dealing with multiple lineages or different variants of the virus circulating in given environment. So, but we were able to eventually conquer that after 2 more years and eventually come up with pan-Lassa. That is a Lassa test that can pick up all of the different lineages circulating in the environment. I often joke by saying that, you know, when you do research in Nigeria, you should be prepared to face a lot of the challenges. And we all face it, you know, light, electricity, and everything. But I said, apart from light, electricity, and the environment, even the viruses here are very difficult. So you really have to think, you know, a bit more differently the way you work here compared to when you work elsewhere. So we really came up with this, and then this seminar work gave us this cell, cover, and that has really accelerated the way things done. We started in 2015, and between 2015 and 2020, that accelerated the development of novel vaccine, and now we have 2 vaccines in which we involved in clinical trial in Nigeria. So now the other study I will be talking about has to do with the Lassa fever surge that we had in 2018. So in 2018, Nigeria's, suffered what I call a major surge of the virus. That is, in 8 weeks, we had more cases and more death in the country than we have in the past 2 years combined. There were a lot of unanswered questions. Were we dealing with a new variant? Was this sustained by human-to-human transmission? Was this due to a virus that is different or the diagnosis not working? So we had all of these questions. And I remember there was a lot of panic in town. In some instances, they even say Ebola was back because we were having many people dying, and the disease also manifests like Ebola. We're bleeding through all the orifices. And we had, as a member of the National Technical Working Group on Viral Hemorrhagic Fevers, I do remember we had a meeting in Abuja between the chairman, Professor Oyewale Tomori, and the Minister of Health at the time. I was told to solve the problem. We're trying to think about it. They said, look, you have everything, go and tell us what is responsible for this problem. So we went on and then collected samples across, you can see here. And then within a week, we were able to come up with a report. And that report was very simple. We found that there was no evidence of a particular new viral strain, no evidence of increased human-to-human transmission. But what we found was it was multiple introduction of the same variant that we know, from animal reservoirs to human. And that really helped us to understand a lot of things. We identified a few clusters through nosocomial transmission from patient to clinical personnel. And those clusters were between 2 to 8. And that also made us see one thing very clear. During the outbreak, we discovered that the lineage 2 that we knew before has divided, and then we had lineage 2B in the east, 2A in the west, and then lineage 3 in the northern part of Nigeria. So the country was basically dealing with with 3 different epidemics, but all compounded into one. And that for us was very instrumental because looking at these natural borders, we realized that they were all clustered and then divided around the natural geographical borders, the Benue River, the Niger River in Nigeria. And that made us understand that it was mainly about rodent. And at that time, we had no skill, no expertise into dealing with research using animals. But I was very fortunate that I had, as my companion, my better half, and my wife, a veterinary pathologist. So I just turned around and I called out my wife. I said, well, look, you need to come and help. We don't have the skill. And that's where she really agreed to come help us and set up the whole program, the zoonotic research program that we're enjoying today. You could see her here in the field. We went to the field trapping rodents in household of people that had Lassa fever. And this is the very first demonstration at the time. From the work that she did, collecting samples from people that had Lassa fever and collecting rodents in the household of people that had Lassa fever, we demonstrated for the first time that you see the same virus between the animal and then the human. And that is the very first time that you could see that demonstration using whole genome sequencing, and showing that that really is the thing. But beyond that, she also helped us to do something that was unprecedented. She started looking into other animals around the household. I am not a vet, and I can't think the way they think. I really have to give it to them. Veterinarians are very smart. And she really was able to show us one thing, that the virus that we think about being only in rats is not only in rats. That virus is also in other domestic animals. And therefore, the lesson is we have to be worried. We shouldn't just say Lassa fever is found only in rats. It's also found in domestic animals. And this really is something that she recently published, showing that really the reservoirs are many more than we ever thought. What we did at this time is that we also adapted and we adjusted. Remember, we developed a rapid diagnostic test a few years ago. Yes, it worked. But then the antigen capture test that we developed also has some limitation. Anytime you talk about antigen capture tests, they will tell you sensitivity, specificity, is not a nucleic acid-based test. We talk about cross-reactivity. A whole lot of those things come up. So, but we leverage, the evolution, the advent of the gene editing technology, to develop what we call the CRISPR. We developed the advent of CRISPR technology to develop this test called Sherlock, which basically just enable us to detect Lassa fever, but detecting it in a small test tube that will cost you about $3 instead of $130, and you don't need any equipment. That is, what we did here through this whole process was to convert the whole process of PCR, but then render that PCR, and turn it into an assay that you can do on a sheet of paper. So imagine you are doing the pregnancy test, but then you are putting everything, your enzymes, your nucleotides, everything on a paper. And what you have to do is to stick it in your mouth, get on saliva, or just urine and get it in a tube, and you can get the result in 10 minutes. And it's a nucleic acid test. By the time we did a comparison with PCR, it's neck to neck and 100% sensitivity and specificity. So, and then this test really has been very revolutionary. So basically, using genome sequence data and then creating guide RNAs, and then identify the guide RNA, use nucleases, molecular scissors to cut, and then have a reporter. And then the kind of result you get is this. Positive, negative, but what is important is you are doing a PCR on a sheet of paper. You don't need a thermocycler. You don't need to be educated to do it. You just have to get saliva or urine, and then you get the same result that you can get on a PCR test. When news of this assay came out, Nature could not believe that, because this is that was the very first CRISPR-based test on any infectious disease. So they sent one of the writers of Nature in our lab, Amy Maxson, and she was in our lab for a whole week because she wanted to confirm that this thing is done in Africa, is done by young Africans. She was there for the whole week and saw the process from the beginning to the end. And then she went on and wrote this piece in Nature, Faster, Better, Cheaper, the rise of CRISPR in infectious disease detection. So, we basically were not looking only at the disease. Yes, we'll conquer the virus. But then when you are in the field, you encounter quite a number of challenges, especially working with clinicians. You are being asked questions that you cannot ask. I remember one of our lead clinicians in Irua would tell us, well, you have all the diagnoses, they are working. But we have some patients that come here, they have symptoms that are very similar to Lassa fever. Your tests are negative, and then when we treat them, at times they respond, at times they go on to die. There is a problem with your test. There is a problem, maybe you're thinking, maybe we have other viruses that are responsible for the disease. And then the other question that I was being asked is, how come in the same household we have somebody that has Lassa fever? His situation is very bad. He goes on to die. You have another person that has his own, is very mild. Or you have somebody that is exposed and is going about without disease. Some of them are asymptomatic carriers. You look at the community, it's the same picture. So the question was, what makes some people more susceptible to Lassa fever than others? What makes some people more resistant to Lassa fever than others? And that really started making us thinking that there could be some human genetic determinants that are responsible for the outcome that you observe. And we basically took advantage of a program called the 1000 Genome Project, where we were involved and then we basically were responsible. If you look at this map over here, we collected samples among the Yorubas, the Mendes of Sierra Leone, and then the Ishans of Nigeria, where we were working on Lassa fever. This project is a global project. We had the human genome sequence in the year 2000, but we all understood that technologically it was challenging. Now we wanted to establish the new human genome, taking into account advances in technology. We work on these three populations, and then what is interesting about the fact that the three populations on which we work, that is the Mendes of Sierra Leone, the Asians of Nigeria, and then the Yorubas, are the population with the highest number of genetic variation in their genomes. And the question is, why? You'll get the answer afterwards. It's basically, I can start telling you, most of the genetic variation that you see are driven by infectious diseases. So we went on to do this, looking at doing genome-wide scan. That is basically collecting samples, mother, fathers, and then children, and then really conducted what I call genome-wide scan. That is sequencing the whole genome, and then looking into the differences into the genomes of people. And what we found, is that if you look at, we identify you could look at this area, quite very high in the Yorubas of Nigeria. If you look at it in the Asians of Nigeria, it's a bit low. And then you look at the Mendes of Sierra Leone, it's actually lower. We really started focusing in this area. Why are we seeing this level of polymorphism? This, what I call signals of natural selection in these different populations, and why did they differ? We zoom into that area, into one particular gene called the large gene. And this is what we found recently. This paper is coming up in Nature. It's been accepted. So we found, for instance, one particular set of mutation on chromosome 22 and in this locus into the large gene and in the Nigerian population that makes them, that this is what absolutely protect people in this part of the world. So, and our hypothesis is we have a very strong hypothesis now about this. And this is what is driving our novel research on identifying the new treatment for Lassa fever, because this disease actually doesn't have a treatment on its own. So we believe that large, which is a receptor on the surface of the protein, after dystoglycan, which is a receptor of the protein, is responsible, after dystoglycan here, is responsible for bringing the virus into the cell. But for dystoglycan to function, you need to recruit the large protein, which is the protein that is encoded by this gene, and that has some mutation that can protect against Lassa fever. So this large protein glycosylates, alpha dystoglycan, and then is actually sent on the surface of the cell. And it serves as a receptor to bring the Lassa virus into the cell. But once you have mutations at large, then the receptor ligand, receptor virus ligand interaction is actually affected, and then that blocks the virus to come into the cell. So we've done in vitro and functional analysis, and then we have it very clear that those mutations, when they are present, you get exposed, you get infected, but you don't get sick because the virus does not find its way into the cell. And this is our latest finding on this, and we believe that now we've actually identified and uncovered the mechanism of resistance to Lassa fever. As I mentioned before, when you are dealing with the clinical team and then they are telling you we have viruses that are present, and then they see patients that have clinical symptoms of diseases, all your tests are negative. The question is, what is responsible? So to answer that question, in 2015, we conducted actually the largest clinical metagenomic study in Africa by collecting thousands of samples from people who had fever and those that did not have fever. We sequenced those samples, by next generation sequencing, with the hope that we can actually identify the different pathogens that are responsible, and then through that we can actually develop countermeasures, which vaccine, drugs, or therapies. You could see here, looking at healthy and then febrile patients, what is interesting is the fact that we discovered 2 novel rhabdoviruses. And to our surprise, these novel rhabdoviruses that we expect, if you look at these numbers, for instance, to be in this group, you rather find them in this particular group here. So you find them in healthy persons, in quote. How did you find this in healthy persons? We went back and then realized that they were taking traditional herbs. And these traditional herbs reduced the pyrexia at the time, and then they appeared to be healthy at the time. And then by the time we did this assay, we found that 80% of people, we developed an ELISA assay, and we found that 80% of people in those communities had exposure. So these 2 viruses that we called epomavirus 1 and epomavirus 2, and they were responsible, they are similar to the Bakongo virus that was responsible for viral hemorrhagic fever in the DRC in 2012. So, and that for us was very revealing, that you could use this as a way to discover 2 novel viruses. This is another case study that was referred to. In 2018 November, within a week, Nigeria lost 179 school children around Edo State and the surrounding. What was responsible for those number of deaths was not known. They tried all kinds of tests, malaria, yellow fever, Lassa fever, with the tests that are available. All the tests were negative. The Nigeria Center for Disease Control sent 22 samples to our lab for testing. And we just went on and then performed metagenomics. We found that 20 of them were full genomes of 20 of those samples, and those genomes were all yellow fever. But the difference between the yellow fever that we found in our lab here, these are the previous yellow fever samples found in Nigeria. This yellow fever here that we found all clustered together, but they were different from the ones that have been circulating in Nigeria in the past 94 years. This information was given immediately to the Nigeria Center for Disease Control. They declared an outbreak, and together with the National Primary Healthcare Development Authority, they were able to contain that outbreak within 2 weeks, saving thousands of lives. This is another use case in a hospital where we work in Lagos called Ikorodu General Hospital. A mother and a father present with a two-year-old baby that is bleeding from all orifices. Nose, ears, mouth, everywhere. Eyes, bleeding. They bring the child to the pediatric emergency ward, and then the emergency and the pediatric ward just empty. They all disappeared. Everybody just ran away because the notion that Ebola was back. Our team was called. We went to collect the sample 48 hours. We found that it was actually a heavy load of enterovirus B. The result was given back to the hospital. That built confidence, and then they all went back and then started managing other patients, even though this child along the line, because of neglect, died. 2021, in the midst of the outbreak, we have a case in Lagos where patients are being treated. A TB patient treated with antibiotics for TB. TB treatment using the conventional method, found that these patients have MDR-TB. But there was a question. These patients were not responding, and then their cases were really bad. So the sample got to a lab, and I got 2 of my graduate students working on this, and they were able to establish the very first case of extensively drug-resistant tuberculosis in Nigeria and in West Africa using microbial metagenomics. This information, again, was shared with public health authorities so for them to be aware of what is going on. As I said before, we have changed gear and really look beyond just one human. So now we're using a One Health approach to address this problem because we realize that you have spillovers from animal to human. And I'm glad to say that my deputy director is actually leading one of the largest zoonotic surveillance in West Africa supported by all these organizations. And we've been able to find a lot of things. One here, you could see them in the field here. A few years, in 2021, we had a case, the Ebola of rabbits. It happened. Rabbits dying, decimating farms are all decimated. And they are bleeding from all orifices, nose, eyes, ear, everywhere. What is responsible? Samples were taken down to a lab, and together with a team of young veterinarians, they were able to establish the very first rabbit hemorrhagic disease virus genome in sub-Saharan Africa. What is funny about this story is that when this outbreak occurred, it went global. The US Department of Agriculture reached out to the Africa CDC, and they wanted samples to be sent to America for sequencing. And the Africa CDC made them understand that if it is in Nigeria, and then those samples are available, I'm not sure you will have access to samples. You may eventually have access to genome sequence data. So by the time we had the video conference with them, they were asking for samples, and we told them that no, you can have access to samples. The samples, we have the data. So the sequences are done. And that led to a very fantastic collaboration. So we're basically not just looking into animals, we're also looking into mosquitoes. And then we've really found a very major breakthrough recently by looking at mosquitoes in environment where they had yellow fever to see what are those drivers of those pathogens that catalyze the yellow fever virus in mosquitoes. And that really showed us a diverse number of viruses, but I want to spare you from the detail. A very interesting paper. We've also looked into bats, and we basically just recently identified novel beta coronaviruses into bats, and then this is really driven by some of the young students in my lab or graduate student. But what is interesting about this is that the novel coronavirus that we discovered has been very important. It's very important because it's broadened our understanding of this virus evolution, and that can help to actually broaden the spectrum of the kind of vaccine that should be developed for coronavirus. And recently, we just went into and used monkeypox sequence data actually to show that the outbreak that we have in Europe now is not emerging or emanating from Africa. We use this absolutely to bring WHO back to order. The reason being that what is circulating now is a completely different clade. But they are trying to link it back to Africa and especially to Nigeria. But we use the data to convince WHO and let them see that they really have no understanding, and that whole appellation linking these viruses sent to West Africa should stop. It is a completely different strain. It's a lineage circulating here. This is what is circulating within Africa. And then this is the data that will actually show that this is a different cluster. So, and they will also ask them to stop, using the wrong nomenclature. Remember that monkeypox actually as a virus was described for the first time in Denmark, but it doesn't carry the word Denmark into it. COVID started in China, but it's not called China pox or China virus or anything else. But we are very good at calling things with African names. Here we're talking about Central Africa, and we call it about West Africa, and then basically narrow it down to Nigeria. But we've been able to actually make them understand that this has to change. So when you hear WHO saying that they are changing the nomenclature or the name of monkeypox, they didn't change it because they like it. They were forced to change it using scientific data. So all of this that I mentioned before has really led us to set up this program that I mentioned here. pandemic, preemption response is built on 3 pillars. Early detection, as I mentioned to you, using next generation diagnostics, creating multiple diagnostics, and then also setting genome centers, passing information in real time to public health authorities, and also empowering frontline healthcare workers. So it is basically built on these 3 pillars. So hub and spoke models, where you have a genomic hub that has a lot of spoke around it, and then you can empower them to do a lot of things. And then you connect using data science or information technology to share information in real time, and then also empower the workforce. We believe that through that, you could actually just expand the scientific capacity and enable sustainable independent surveillance program on the continent. But then this is what this program had to do. Early in the pandemic, you remember, COVID was first found in Egypt, in Africa, then South Africa. Nigeria was the 3rd country to report COVID. But interestingly, Nigeria became the first country to do the first genome of COVID in Africa. It's simply because we pulled out the big guns. We set up the technology, we set up the platform. We got the sample from NCDC, and within 48 hours, we were able to show that this sample was not due to internal, what we call local transmission. It was basically basically acquired, or the person was infected in Switzerland, to be more precise. Even though he was an Italian, he got his infection in Switzerland, traveled to Nigeria, and then was found sick here. And that was the very first SARS-CoV-2 genome in Africa. But then this really helped the continent to understand that they could do it. They could understand that we could move beyond the barrier and then do things better. What is interesting is the fact that we did this in 48 hours. This is a speed that up to now, most countries in the world can't do. And the question is always, how do you do it? Because for an outbreak, you need 2 things, speed and accuracy. So we went on to be one of the second group in the world to identify the very first variant of COVID after the Alpha variant with this D64G mutation. When we found this, we were able to relate that with the numbers increasing over time. And then this was really related to the first surge that you see. This information we use to share them with public health authorities. And I can tell you that the Nigerian government, the Nigeria Center for Disease Control, could not do anything. Every Monday, when you hear about the Presidential Steering Committee in Nigeria, that meeting could not hold unless we send the data, because they were working based on data. You could see here, over space and time, this is kind of dashboard that they had. And you could see spreading, this is developed in our lab. Over time, we knew what virus was circulating and what was going on where. At some point, we knew that 49 different lineages or variants were circulating in Nigeria. We knew the different places. And we used to send a report to NCDC every Saturday or Sunday. If we don't submit, then they don't have a meeting. So in real sense, when you hear the former director of NCDC, Dr. Chikwe, say, response in Nigeria was guided by science, yes, it was guided by science. It was quiet, but they had all the scientific evidence to make the decisions. And we just kept having the map, showing all of this map. But what is important is the fact that you could see over time, you see new variant, new lineages coming in. One coming in, taking over, disappearing. The other one coming in over and then taking over, disappearing. So these were the signs that were guiding the response. And apart from looking into Nigeria, we're basically also tracking what was going elsewhere. Because the idea was to track and then see what you can track to prevent. And then this is a paper here by Dr. Happy, and one of our former students, where they really, again, used genomics as a way to start tracking the emergence of SARS-CoV-2 variant in South Africa and see how that were moving. And this was published in Nature Medicine, but actually showing how you can apply this as a way to control an epidemic. What we did, within 8 weeks of COVID in Nigeria, we developed this, again, CRISPR-based rapid diagnostic test. And this test, this is the very first test that we developed here, very raw, and then we went on to create, this is a power-free. So basically, this is how it appears now. So whereby you could get this within 15 minutes, and then you can get your color, it appears. So you just get your saliva in, and then you can get this test done. This test was developed in this country. The reason why it's not here, don't ask me. But it's done elsewhere. With all of the tests and all of the things that we've done, we've now gone and created a novel technology that is revolutionary, and it's called MCARMEN. It's a multiple combinatorial array of multiple CRISPR tests, whereby now we have the ability to combine up to 469 different pathogens in one single test. And you could do that test and detect it in one single assay. It's all about moving beyond the boundary, technology. So, and then you could do this in multiplex. So more or less, you know, one-stop shop, whereby with all of the work what we've done, we should be able to combine all of that in one platform, and then we can have them done. Interestingly, as far back as September 2020, we had gone on with all of the sequences that we've done to create a candidate vaccine for COVID-19. But that's where I realized that science, politics, and geopolitics could be very important, because we had a vaccine that was more efficient than what Moderna and then Pfizer had. We had a vaccine that had the spread. You're going to see the next one. And it was all based on genomic sequence data gathered on the continent because we supported them. Basically, going from genomic sequences, we use bioinformatics and machine learning to remove all of the epitopes, you know, and they call them antibody-dependent enhancers. And those are the ones that are responsible for cytokine storm that make people very sick. You get in the epitopes that really make the person safe. So we put them into our construct, did an in vitro screen, and then from the in vitro screen, we pass them into animal model. And we're able to show an efficacy of 95% at a time in animal model. Now, that vaccine that I mentioned, we took it up to the highest level in this country. But for a reason that I don't want to talk about, it was not supported. But another country, very small in Africa, took that vaccine, and that vaccine is owned by a country called Botswana today. After doing a clinical trial in the UK, the vaccine is going phase 2 clinical trial there. And I'm very sure that at some point, we'll buy the technology from Botswana to apply that in the country, and yet we have the ability to take it. So basically, this is just to show you how spread in terms of protection, because we didn't just focus on SARS-CoV-2, we actually focus on different coronaviruses to make it broad spectrum. And you could look at the neutralization data here, very high. So in terms of neutralizing the antibody, we had it all covered and well protected. We also using all of the skills and knowledge and technology that I mentioned to train. We've trained over 1,300 people in short term and then master's and PhD program, and then we're really partnering with public health institutions. And then this gives you the map, and this tells you this is how much we've impacted Africa. We've trained 42 different African countries out of 54. And this has a tremendous impact on the way Africa responded to the epidemic. You can see the map of Africa is fully covered. From a small place here in Ede, we've been able to reach out to the whole continent. But what is important is the fact that this training and the trainings and the technical support that we gave resulted in this. In 2020, we had barely 5,000 genome sequences. But because of the training we provided, by around August, we're over 100,000 sequences, and that number actually just exploded after this. So we had those 2 papers in Science that we published to show the impact of this, you know, in the first year, and then how this has really transformed the way Africa responded to the epidemic. And I can tell you that the science was so powerful that countries actually understood the value of genomics. A country like Rwanda, I've freighted an aircraft with thousands of samples from Rwanda down to Nigeria. The samples were sent to us. We did a sequence because they had a number of cases exploding. They didn't understand what was going on. And we, within a few days, did all the analysis and mapped out, and we realized that at that time, Rwanda had 97.3% of the Delta variant. And we had a conference call with the government, and we recommended lockdown in those hotspots. And then they went on a hard lockdown of 3 weeks. By the time the set of samples were sent back, they have gone from 97.3% to about 16.9%. actually showing how you could use this technology to respond to an epidemic. That helped a lot in Africans discovering all these variants of concern, variants of interest, and then also seeing how this was migrating within the continent. That really shows the firepower behind this technology and see how you could use it to contain outbreak. Now, what is important about this, unfortunately it's not very clear, is that we've created a dashboard. And this dashboard makes data in real time, whereby you have a test somewhere, anywhere in Nigeria, then the minister can see with GPS data the type of test that was done, and then actually, and I can use that information to create, to create an early warning response. So this is the case that we create for COVID, and this is what the ministry and the presidential task force was using, where you could use this. They don't have to be genomics. It was all about clicking on the button and you see all the different variants. You could see the history, how it moves up, how it comes down, where are the hotspots in Nigeria. And this really is what has been driving the response, not only in Nigeria, but we have something like this at the Africa CDC level. And this is what the African governments are actually using to respond to epidemics. So I really want to end by really thanking everyone and thanking my team, and especially thanking those who have funded us to do all this work. Thank you.
Another round of applause for Professor Christian Happy. Can we have members of the high table back to the high table, please? I apologize for the quality of the audio. Online, it was crystal clear. That was why I had to join online myself. It was crystal clear. I think we'll like to have a recorded version sent to all those who have their emails. If the foundation can do that, we'll appreciate it. Distinguished guests, ladies and gentlemen, permit me to hand over to Professor Grace Botosha, who will then handle the next session of the program. Thank you.
We want to say a big thank you to our distinguished lecturer. That was a fantastic presentation. Please, and fantastic work that he's doing. I would call it translational research that is actually impacting public health. Congratulations and well done. Can we celebrate him once again? And so we'll like to call the Vice Chancellor, sir, the Chairman of the Thomas Bassea Biomedical Foundation, who is being represented by one of the members of the Board of Trustees, our immediate past Provost, Professor Olaokpa, Olubumi Olaokpa. And I would also call Barrister Oyinda Bassea, daughter of Professor Olumbe Bassea, to make a presentation on behalf of the board to our distinguished speaker. Please, could you rise, Professor Happy, while this is done? Okay, just stay where you are. Yes, I think you should.
The Bassea Thomas Biomedical Foundation. On behalf of the Bassea Thomas Biomedical Foundation, we present you this. Thank you very much, sir.
Could I just say that what Professor Happy did not tell us is that he came as a two-in-one. Your presentation showed that Dr. Happy is a very major contributor to the work. We congratulate you and we celebrate you. So that should read and we say, yes, that's the proper way to do it. Thank you very much. Just before the chairman speaks, again, we apologize for the quality of the audio. As has been requested by the Master of Ceremonies, for all those who we have their email addresses, we will send the link to the YouTube so you can watch the lecture without the speaker problems. We apologize.
Distinguished guests, ladies and gentlemen, permit me to call on the Vice Chancellor of the University of Ibadan, who is ably represented by the Deputy Vice Chancellor Administration and who has chaired today's ceremony, Professor Ezekiel Ayola, to make his closing remarks on this occasion. Thank you, sir.
Thank you very much. Once again, on behalf of our Vice Chancellor, I want to thank our guest lecturer for that beautiful and wonderful presentation. Even those of us that are not in the field, we can follow what you are saying. And if you agree with me that it's a lot of work that has been done in the remembrance of Professor Olube Basia. Once again, we'd like to thank everyone that is here, those who are online and those who are physically present to grace this occasion. We appreciate you all, and we thank you very much for your patience and for listening to the lecture. On behalf of the Vice Chancellor, once again, I'd like to thank you, the organizer, the local organizing committee, and everyone that have been involved in this activity. So, on behalf of the VC, I thank you. Thank you very much.
Thank you so much, Professor Ezekiel Ayola, Deputy Vice Chancellor, Administration of the University of Ibadan. It is now my pleasure to invite to give the vote of thanks Professor Grace Gotoshaw. Thank you so much, ma'am.
Thank you very much. So on behalf of the chairman of the board of Thomas Basia Biomedical Foundation and also members of the board, we want to appreciate deeply the Vice Chancellor of our great university, Professor K.O. Adebowale, who has been ably represented by the Deputy Vice Chancellor Administration, Professor Ezekiel Ayola. Thank you so much for supporting us. Thank you for being here on time. Thank you for taking on this assignment, because as at yesterday, I was communicating with you. We understand that the Vice Chancellor is in Abuja, and I was also able to communicate with him. And he said Professor Ayola will be representing him. Thank you so much, and thank you for supporting the Thomas Basia Biomedical Foundation. I'd like to also thank all directors that are here. We did have the director of the Research Management Office, Professor Jegede, was here for some time. We are grateful that he was able to spend time here. We appreciate all heads of departments. I'm sorry I can't recognize each and every one now, but permit me to recognize Professor F.A. Feintola, Head, Department of Pharmacology. Thank you so much for coming. We want to appreciate all our distinguished professors, both retired and not tired. And just please permit me to appreciate Professor Atinmo, who was here very, very early. Thank you so much, sir. We also appreciate our retired professor, but not tired, Professor Ekundayo. Thank you so much. He was also here early and he has sat through the entire program. We appreciate you. All distinguished professors that are here, we'd like to also appreciate you. Permit me to also appreciate our own darling mother, retired but not tired professor, excellent researcher, major award winner, Professor Adeyinka Falusi. Thank you so, so much. It's so nice to have you in our midst. She's always at all Thomas Basia Biomedical Foundation programs. Thank you very much, ma'am. I want to say a big thank you to all the academic staff that are here, other professors and other lecturers, associate professors, readers, senior lecturers who are in our midst. Thank you very much for creating the time to be here. I also want to appreciate all the postgraduate students. I'm sure you've learned a lot. And I think I can say that at the end of this whole session, please feel free to speak with the distinguished lecturer and his amiable wife, who is also an excellent researcher. So please feel free. They will still stay around if you have questions, if you have areas you think they can help you out with, or you even want to collaborate. I'm sure they'll be most willing. I'd like to say a big thank you to the members of the late Professor Olumbe Basia family. We had the barrister, Onyida Amola Basia, present the plaque, and she's here with her daughter, that is the granddaughter of Professor Olumbe Basia. Thank you so much for standing by the foundation. I'd like to also, at this juncture, thank our distinguished lecturer, Professor Christian Happy, and his amiable consort, who has become a co-collaborator with him. Can we celebrate Dr. Anis Happy? Christian, I am not disappointed, and I'm so proud of you. At the strides you are making, at the impact. You've always had a passion for research and a passion for finding solutions to human health problems. So I'm not disappointed, I'm not surprised. I know the sky is not your limit. Well done, well done, well done. Thank you for what you are doing in Nigeria. And let me just quickly tell you, he's not Nigerian, he's Cameroonian. And yet he is doing so much for us in Nigeria. Can we celebrate him again? Thank you so much. I want to thank the online audience. They have also sent all sorts of accolades. Excellent work, amazing work. The Vice Chancellor of Christian University, Professor Babalola, put on the chats online, wonderful service to mankind. Professor Farumbi also said, excellent presentation, excellent work, amongst other comments online. Thank you so much for heeding this call and for doing us so proud in research and also in Nigeria here. Of course, thank you also for doing the Cameroonian people proud as well, because I'm sure they are so proud of you. Once again, postgraduate students, I appreciate your presence here. We want to say a big thank you to the members of the press. We have the journalist from Tribune here, Mrs. Shade Oguntolu. Thank you so much for coming. And for the ITU from the College of Medicine, thank you so much for covering this event and live streaming it. The photographers, we really appreciate you. And all those who have been working behind the scenes, we say a very big thank you. Once again, thank you very much for coming, and God bless you.
Thank you so much, Professor Watershaw. A quick announcement is that immediately the program ends, there will be photo shoots outside the venue, and to be for members of the high table first, and then with distinguished professors and teachers in the house, and then the entire audience. We'll find a way of taking snapshots of those online. Perhaps the IT can ask them to show themselves and then take a shot as we close. On behalf of the Thomas Basset Biomedical Foundation, we close this program. Thank you so much all for coming. Safe journey to your respective destinations. Bye-bye.